Hello, everyone, and thank you for joining us today. I'm Sean Fensky, Editor-in-Chief of Medical Product Outsourcing and moderator for today's program. You've logged on to the MPO webinar, Package Validations, a look at current and future state testing. Before we get started, I have a couple of housekeeping announcements. For those of you who are interested in the information you hear today once the webinar has ended, or if you'd like to forward it to a colleague, this session will be archived for a year. The archive will be able to be accessed through the Medical Product Outsourcing website, which is at www.mpomag.com. Also, we will be accepting questions throughout the presentation. You can type your question into the Ask a Question box in the webcast window. We will collect all questions and hold them until the end, but please feel free to type in questions as you have them. And now, before I introduce you to today's presenter, I'd like to take this opportunity to thank our sponsor, Nelson Labs. Nelson Labs, a Sotera Health company, is a global provider of laboratory testing and expert advisory services. The firm performs over 700 rigorous microbiological and analytical laboratory tests across the medical device, pharmaceutical, and tissue industries. Nelson Labs recognizes that every test matters and requires solutions to complex problems to improve patient outcomes and minimize client risk. And now let's get started. Today we're joined by Wendy Mach, Consulting Manager at Nelson Labs. Wendy has over 24 years of medical device manufacturing and laboratory experience. She worked in the medical manufacturing environment in Minnesota for 7 years, performing ethylene oxide and gamma radiation sterilization validations. While at the manufacturing site, she also insourced the microbiology test for sterility assurance and related equipment validation. Wendy also spent 4 years as a lab supervisor managing the sterility, microbial limits, bioburden, endotoxin, and antimicrobial effectiveness services. She joined Nelson Labs in 2004. And now, beginning her presentation on package validations, is Wendy Mach. Wendy?
Thank you, Sean, and welcome everyone. Sorry about that. Two-finger clicky. Okay, here we go with the agenda. Today's webinar is focusing on current and future state testing of validations. We're going to go through the purpose of packaging, and then I'm going to touch on some applicable standards. One of the most interesting parts is going to be the medical device regulations and what changes they are driving in the standards. I'm going to go through some common test methods, and then I'm going to talk a little bit about sample size considerations. So let's get started with this exciting stuff. Okay, what's the purpose of packaging, or the sterile barrier system as it is known? The sterile barrier system is designed to allow necessary sterilization. The important parts about that is if you're using ethylene oxide, you need to ensure that the material is porous or breathable, allowing for the penetration and removal of the gas. When using radiation, you need to ensure that the compatibility between the materials and the radiation is applicable. There are a lot of effects that can happen from radiation, and so you need to ensure that they are compatible. The third area is steam. Steam is a very high temperature and water vapor, so ensuring that these temperatures and water do not have destructive characteristics is very important. Additionally, allowing for aseptic presentation is another key factor, making sure that once you get your samples to their point of use that they can be opened in a manner that maintains their sterilization without damaging anything. And then finally, providing an acceptable microbial barrier. The last thing you want to do is spend all this money on packaging materials that are compatible for sterilization, and then the design that it's openable and it doesn't compromise the product, and then all of a sudden you get to the facility and it's punctured or the seal didn't hold. So making sure that it maintains and is an acceptable microbial barrier. These are the current standards that are out there for regulating the packaging, the GMP 21 CFR is the FDA regulation stating that each manufacturer ensures that the device and the containers are designed and constructed. The second one is the one that most medical devices and pharmaceuticals, all of those terminally sterilized products have to follow. This is what I refer to as the packaging Bible. 11607 has recently just been revised, and it is of two parts. Part one is materials and sterile burial systems packaging, and then the second part is the requirements regarding validations for forming and sealing. TIR 22 has been withdrawn and was replaced by ISO 16775. This is also currently up for revision now and is being worked through. The last discussion, I believe, we had over 500 comments, so it's going to be a little bit before that one's finished. Then finally, the last document is EN 868, parts 2 through 10. Part 1 is harmonized with ISO 11607. So compliance to 11607 is corresponding to 868-1. Parts 2 through 10 are specific to individual material types. For example, Part 5 is specific to sealable pouches. Okay, what are the changes for the MDR? Why does it matter? A number of areas, the MDRs have increased emphasis and more explicit requirements. We want to make sure that these are aligned with harmonized standards and industry guidance. Specifically, the 11607, as I mentioned earlier, was revised and released in Europe to coincide with these requirements. The transition date is May of 2020, and in general, there is no grandfathering clause. However, there are exceptions that are outlined in Article 120, if you're interested in whether those apply. A couple of things that are specifically from the impact of the MDR changes is the definition of a medical device. It states that products specifically intended for the cleaning, disinfection, or sterilization shall be deemed to be medical devices. This is an important definition because it leads to the corresponding accessory for a medical device. An accessory is an article which, whilst not being itself a medical device, is intended by its manufacturer to be used together with one or several medical devices, specifically enabling the device to be used in accordance to its intended purpose. This inclusion has placed packaging and product at the same level. Both of those definitions are actually significantly longer, and if you're really interested, definitely get a hold of the MDR changes and read those because they're very long and specific definitions. So I'm going to go through a couple of highlights for these changes that are specific to packaging. And the very first one is related to Chapter 2, Requirements Regarding Design and Manufacture, which is outlined in Section 11. And it states that devices and manufacturing processes shall be designed to eliminate or reduce the risk of infection to the patient. What this is getting at is that a package serves as a means of dispensing the product to the end user, and therefore the design must allow for easy and safe handling while minimizing microbial contamination. To meet this requirement, Section 7 of 11607 outlines usability evaluations. A documented usability evaluation will demonstrate that the sterile contents can be aseptically removed, meeting that easy and safe handling requirement from the sterile barrier by assessing the ability of proper aseptic technique in the intended clinical environment. Let's look at this in a little more detail. Usability. So usability is nothing new to the medical devices. This has been part of the normal design process and is now moving into the incorporation of this aspect into the packaging realm. That is the new approach. These usability studies allow the manufacturers to mitigate risks using objective evidence associated with the correct use of the package. It can also lead to a higher understanding of the factors controlling the quality of user interactions with your product. There were a few approaches to gathering this data. Contextual inquiries is a very common approach that can be conducted to understand the needs of a market and to scope the opportunities through watching the user in the course of normal activities and then discussing those activities with the users. This can be done with one-on-one interviews or with group settings. Annex J.4 of 16775 is an informative reference that provides an exceptional example of attributes that can be evaluated prior to opening by the end user. If you haven't had a chance to take a look at that, it's really good. It breaks it down into things regarding the product, things regarding the packaging. It's a great annex. Let's look at highlight number 2, and I'm not going to read this entire slide. The important part is underlined in that it shall be ensured that the integrity of packaging is clearly evident to the final user. This has been addressed by 11607 by incorporating a best practice visual inspection to demonstrate that the integrity is clearly evident. This also ties into ensuring that if the package is damaged, that you're giving them the what to do's if it's damaged. Do you return them? Is it a multi-package? Is it the outside that's damaged and then therefore you can accept it because the insides? This is also another great area where you can get some feedback on what the market thinks. If you have a double pouch system and the outside is compromised from some process, shipping or something like that, the nurse, even though the sterile barrier system has maintained its integrity for the package and the device on the inside, the end user may not feel comfortable using that. So incorporating those usabilities to address this is also a really good way to address the first highlight. Here's a couple more things that also changed in the 11607 document that were driven from the MDRs. One important interesting point is that packaging systems for non-sterile devices shall maintain integrity. Now, although it's not in 11607, this is a very common question because people have non-sterile devices, but they still use packaging that's sealed using the same processes. So making sure that you're still ensuring that integrity and cleanliness is a requirement of the MDR. Additionally, ensuring that package validation reports are available for the review of notified bodies. A lot of people have told me that they got a citing because they thought that historical actual use in the field would meet the requirements, say for ship testing, or and then it actually doesn't. So Additionally, changes to material design and sterilization are now addressed in 11607. This is really good because it originally was in the guidance document and now has been incorporated into 11607. But anything that compromises the original validation parameters or the integrity of the SBS, such as the introduction of new materials, sterilization process changes, or even possibly a shipping configuration change, all of those items need to be evaluated. It may not end that you have to do retesting, and a possible justification could address the changes, but the act of doing the assessment is what is required. Additionally, the conformity list was updated. This will give you additional test methods on how to meet the requirements of the regulations and whether or not there are established precision and bias statements. And then there is a discussion section on identification of the packaging levels using symbols. Now, this has not been decided. It is just a discussion, but it is moving in a forward direction such that if you have multi-package systems, it will become easier for the end user to know exactly where that sterile barrier system is, whether it's an outside package or an inside package. Okay, let's look at a basic validation plan. At Nelson Laboratories, we do our basic validation plan under a life cycle approach, and that's what this graph represents here. We do all of our ship testing prior to our performance and stability tests. One of the things that I do recommend is that you do prototype tests of your packaging because we've had a couple instances where the client has put a device in a tray, and then we went through the simulation testing for shipping, and the device came free from the clips in the tray. So if you are possible to send a prototype, just send it around and have it come back and take a look and see if your design is moving in the right direction so that you don't have to go back to start. Another key point is the sample size determination. It needs to be statistically valid justification. The historical act of picking a sample size of 11 or 30 or 33 because that's what everybody does is no longer an accepted practice, and the decisions behind why you select your sample size need to be documented as part of those validation plans. Now that you've got your prototype set up and you've established your statistically valid sample size, you're ready to start testing. We do, after our ship testing, we look at baseline testing and we perform 3 tests. We evaluate strength, integrity, and microbial barrier as outlined by the standard. And I'm going to go through one of these tests in depth as we go through this webinar. The important part is that you're going to look at a minimum of 3 points. You're going to do baseline, which will determine whether there was any issues for your samples after ship testing. Then we're going to look at an accelerated aging time point. This is the optional aspect of establishing your shelf life. And then finally, real-time aging, which is the requirement. It is important that all 3 of these aspects start at exactly the same time. There was some situations where clients were just doing their accelerated aging and not doing real-time aging, but that is not acceptable. Real-time aging is the requirement, just to reiterate. Okay, so this is my packaging test summary. These are the most common test methods to meet these requirements. The strength test, you can pick seal, burst, and creep. Each one of these has different aspects. I'm going to go through the seal peel test. For integrity, you have 4 choices. Each one of those is different, dependent upon whether you have a rigid container, whereas you would use the dye immersion, or flexible, which is where most of the other test methods will come into. I'm going to show you some dye migration examples. And then finally, the microbial barrier. The most common test method out there is F1608. This has been around since the 80s. There is a whole package aerosol challenge test, and then F2638. I'm going to go through the microbial barrier properties test, F1608. Okay, let's get started with the strength test. So as I mentioned, the seal peel test is regulated by ASTM F88. You use a 1-inch coupon, which is across the seal, not into the package. And it's very important that you identify the materials in each grip because it can have an effect on the results. If you're comparing your results to, say, an independent laboratory or a lab-to-lab facility, and you're finding significant disagreement between the results, take a look at which materials were in which grips. The most rigid material should be in the mobile grips, whereas the flexibles maintained in the stationary. This can have an impact on the results, so if you run into issues, take a look at those kinds of things. There are a couple options when you're doing the seal peel test listed in the standard. This is the 90-degree unsupported. It is the most common and offers the most conservative value or the lowest. The way that it works is the Instron or tensile machine grips the 2 legs and will pull apart, allowing the tail, which the red arrow is pointing to freely float in any direction as it's pulled apart. This will give you the lowest values. This option is the 90-degree supported. It is done by using your fingers to hold the tail. It's very important that when you're performing this method that you are guiding the tail and not actually pinching it, because you will change the results when you're following this method. This method is the recommended method listed in the EN 868-5 for peelable pouches, and it actually does have a recommended minimum seal strength in there, just as an informational. This is the 180-degree supported technique. A lot of trays fall into this category. The tail is supported in a 90-degree position, and the plate is there to help maintain that. So this is probably the least common method that we've seen. But is really appropriate for trays. A couple things on seal peel failures. If you overseal, for example, this is a Tyvek pouch, you will get this clarifying of material. It can have detrimental effects to the pouch. Now, I've heard in the past that, well, if it's clear, that just means it's sealed really good, which is really not the case. What happens is you've actually melted the material and you actually do get voids, and you've actually damaged the packaging. So oversealing is a method that is listed in the visual inspection as something that you do not want to have. This is another great example of delamination. I include this because a lot of nurses have issues with this creating fibers in the OR. So if you're having delamination issues, corrections to your sealing process would be necessary to ensure that you're alleviating the implication of particulates being released. Couple things, the ASTM is updating. They're looking at evaluating the actual process of not cutting out the coupons from trays. I know that the process itself is very difficult and can lead to variability in the results. Okay, we're going to talk about integrity tests. The integrity test that I selected, which is also the most common, is the bubble emission test. It's regulated by ASTM F2096. It determines the integrity of the entire package and the seal. The way that it works is you submerge the package into water about 1 inch above the surface, and then you inflate it to a validated pressure. The package is then examined for holes or a steady stream of bubbles coming from a single point. This is an example of a bubble emission failure that we received, and it was based on a very common phenomenon from not designing your packaging to fit your product correctly. This customer folded the pouches that were large and pushed them into the shelf box. The bubble test then picked up what seemed to be channels at the fold points, but in actuality, what this was sheet separation or a false failure. And there's a lot of information available on this phenomenon if you're interested. But if this does pop up as part of a failure point and it is at a crease, we recommend that you follow up this test with a dye migration. A dye migration will determine if you really have a true failure or if it is the sheet separation phenomenon. Okay, we're going to jump into the microbial test. The ASTM F1608, as I mentioned, has been around for many, many years, and it is a materials property test that evaluates the log reduction value of the material. This test is only designed for porous materials. You cannot use this on non-porous materials, such as foils. Additionally, you need to have a minimum sample size of raw material of about 47 millimeters. So if you have pouches that are about 2 inches in width, it's very unlikely that this is going to work for you. The standard does have a recommended sample size of 2, and the way that it works is we take these swatches of material and we expose them to an organism, and this organism is actually the very same organism that is used in the ethylene oxide sterilization validations, Bacillus atrophia, so it's a spore former. Upon being exposed to these organisms, we then remove the samples, and the filters that are laid beneath the material are blended and counted, and we determine the amount of organisms that passed through the material. This value then calculates out to what's called a log reduction value. The log reduction values can vary anywhere from 0 to 6.1 or 6.2. The higher the log reduction value, the better retention of the materials. It's very important to note that with this material, we are And with this test, we are using a high flow of air that will drive the microorganisms through the material. Um, I've had a couple people get a little concerned that we were able to actually recover any microbes, but it is important to remember that we're using a really high airflow, and that actually drives the organisms through, and so you will get microorganisms through, and that will convert to a value. Okay, let's talk a little bit about establishing your shelf life or stability testing. Accelerated aging, as I mentioned before, is the option to get product to market while you're waiting for your real-time aging to be complete. It's regulated by ASTM F1980. This document is also going to be currently under revision, just for informational. The theory behind it is that for every 10 degrees above the ambient, you half the shelf life. This does not replace real-time aging, and it is based on the calculations and chemistry of Arrhenius. So let's take a little bit more of a look at that. When you're looking at accelerated aging, it is important to justify both your test conditions and your duration. One of the most common things that you'll notice in the ISO 11607 document is justify, justify, justify. You're going to justify your sample size, you justify your test methods, and you justify your conditions. Ambient temperature that the FDA likes to see is around 25 degrees. You can go down to 20. Again, if you're starting to use these values outside of their comfort level values, such as 25, they like to see why you did that. Establishing your aging temperature, the 55 degrees is the most common approach. I added relative humidity here because it's important to note that humidity is not included as part of the accelerated aging conditioning. If you're interested in determining the effects of what relative humidity does on your packaging, that is managed as part of your shipping and distribution and climactic stressing. That is where you rotate the parameters through high and low humidities and different temperatures. It does not belong in the accelerated aging aspect. Another point that I bring up is to make sure that your aging temperature stays between 10 and 15 degrees Celsius below the thermal glass transition point. We've had some samples come in where the client had us age at the 55 degrees, and after 5 years at 55 degrees, we went in to get the samples, and they just disintegrated in our hands. The polymers weren't designed for that high of a temperature, and they lost over 6 months of time because they weren't attentive to that thermal glass transition point or the characteristics of their plastics. The next question that I frequently get is, should I do accelerated aging with or without product? The process of accelerated aging is to look at the effect of the package and the product. So it is important that you do incorporate your product. If your product is super expensive, it is appropriate to use a facsimile device, a cosmetic defects. Those types of items can be used just as long as they're representative. If you have a medical device and then you decide that you're going to put gauze in there, obviously that's not a good justification. But if you have something that is of similar weight and materials and that is just damaged or something, that's a great option to reduce your costs. Okay, this is our most common question that we get. How do I establish my sample size, and what do I do to document it? So this is the document, ISO 11607, specifically Part 1 states that your sampling plans shall be based upon statistically valid rationale. When you go to the other documents, the TIR used to state exactly the same, and that's why I put this in here, because this was a guidance document. What it gets at over and over again is that it doesn't provide you an actual number. It leaves the justification of how you picked your sample size up to you. You have to define it. This sample size is appropriate if you're doing testing by attributes. However, this is for a single lot approach only. So if you're looking at multi-lots, this won't apply. Additionally, the 868 series, several of those documents, like I've mentioned, do have integrated sample sizes that they want you to look at. While you're looking at the sample size from a number perspective, there are other factors that you need to incorporate, like what is the cost of the product. Now, I'm not telling you that if you have a device that's $10,000 that you can automatically use that justification to say that we're only going to test 3 because we can't afford the cost of the product. But you do need to think about the cost considerations as part of this entire justification. Additionally, the use of risk priority numbers is a common approach that leads into confidence and reliability intervals. This is the most common approach that we use at our company. We use a patient risk-based approach, identifying different quality systems and processes that are in place, and then that will drive a risk priority number, which we then correlate to a confidence and reliability level. Additionally, the test that you're going to be using will drive a test sample size. Attributes are going to have a lot higher sample size than variables, and that's because they're usually a go or fail test. Let's look at that a little bit more. So when you're looking at attribute data, you're getting a pass-fail or a go-no-go type of a result. Dye migration and bubble emission are these types. So you might consider having higher sample sizes for these types of results, whereas when you're doing seal peel or burst, you're getting an actual data point that you can compare to a range. So here's a summary slide of some of the upcoming revisions. The visual inspection, so this is your bonus. The visual inspection for S1886 is currently being revised. They're looking at bringing some of the annex items into the actual document as required inspection points. Currently, the only required inspection point is to look for channels. We're hoping to bring in possible tears and punctures if that is balloted and approved. We have a new standard on determining the minimum seal strength, so there's going to be some guidance there. Like I mentioned before, accelerated aging is currently under revision, and the bubble emission test is also going to be under revision. So there's lots of exciting stuff going on behind the test methods. Additionally, a newly released test method validation document has been released. So if you're interested in some guidance around that, it's a very in-depth approach. Okay, so let's summarize everything that we've looked at today. The MDR changes are driving the updates to the standards. There is definitely a renewed focus on the prevention of microbial ingress, making sure that you've addressed those microbial barrier properties tests. There is the addition of the incorporation of the usability studies. So they've been historically used as part of the medical device, and now they're being incorporated at the packaging level. And then we talked a little bit about the incorporation of the revalidation. When you're looking at different packaging validations, it's very important to make sure that you do address all three properties, strength, integrity, and microbial barrier, and that you evaluate them through the establishment of your use by date. And then finally, we did touch on establishing a statistically valid rationale and some of the approaches that you can use to put that down and get it documented. And now we're going to go back to Sean for questions.
Great. Thank you, Wendy, for that thorough and informative presentation. Once again, don't forget that this webinar will be archived on mpomag.com for a year. Now we'll have the question and answer session, and we'll try to get to as many questions as possible. Wendy, this first question came in very early, the first couple of slides. It is, what is a medical device with, quote, specific microbial state?
Can you say that again? What is a medical device?
What is a medical device with a, quote, specific microbial state?
So if I'm understanding the question correctly, a microbial state being whether it is sterile or non-sterile. So that is where the ISO 11607, that guidance document drives what the requirements are for sterile state packaging. The requirements for non-sterile are, like we addressed a little bit earlier, making sure that you address integrity and cleanliness. I hope that's what the question was asking. If not, please go ahead and submit it in a different manner, and I'll try to take a shot at answering it correctly.
All right, perfect. Our next question is, the microbial barrier testing usually carried out as part of the sterilization adoption / family creation?
The answer to that question is no. It is important to understand that the microbial barrier properties test outlined in the packaging requirements is completely separate and different from the sterilization validations approach.
Okay. This is a little bit longer. If a bubble emission test detects a failure at a fold or wrinkle, can you further explain how the dye migration test can accurately detect a failure, and is dye migration the same as dye penetration?
So the easy question to that is yes, dye penetration and dye migration are the same test. With regard to exploring that, so the way that the bubble test works is that it uses air to find the least difficult pathway to get out. So, and sometimes with the folding of the material, the least difficult pathway will actually be through fibers of the Tyvek material, but that may not be a channel. When you're looking and confirming that failure, you're actually looking at the seal area and if there is a channel between the Tyvek and the film material that's sealed, whereas the bubble was looking for a path through, it's following a tortuous path. So that's where the difference is and why following up with a dye migration would confirm that it's a true channel.
Okay, great. The next question, is it recommended to execute accelerated aging in units previously exposed to climatic conditioning and distribution conditioning?
It depends. So, if you have already verified that the process of the distribution and climactic stressing did not have negative effects on the packaging, so you have proven that it was at the baseline test, then you can proceed with additional samples to the accelerated aging. And I don't have my standard in front of me, but there is a statement in there that although they are separate entities, you do need to evaluate them. So for our approach, we do enforce that the distribution testing did not have negative effects on the packaging after climactic and shipping tests, and if everything is good, then we proceed on with accelerated aging.
Okay. The next question. If a real time for a product is undergoing and cannot be completed before notified body audit for MDR certification, what shall a manufacturer do in this case?
Okay, so obviously ensuring that you have your accelerated aging data available because that represents worst case. But what you need to do is have evidence that the studies are in process. Usually that is in the form of a protocol that outlines how that testing is being managed and controlled.
Okay, we have another question. Is establishing a statistically valid rationale also applicable for non-sterile devices?
Ooh, that's a great question. I would say that if you wanted to get your gold star, that it would be a great document to have in your pocket. Most companies can and do have access to statisticians, and even for validations, which are completely different from packaging validations, you need to have statistically valid sample sizes. So if you're going to be doing that testing, I think that it's always best to ensure that you have that information rather than just picking a number out of the air.
Okay, and we got the follow-up to that very first question I asked you. And his follow-up is whether all sterile medical devices require sterile barrier system.
So I think I'm going to reword that. And the question, I believe, is whether the sterile barrier packaging requires microbial testing. I hope that's what you're driving at. And the answer is yes. The way that the standard is written is that if you can prove that your material is non-porous, and you'd use a Gurley test to do that, then you have met the requirements. However, if you have porous materials, you are required to demonstrate the properties using the tests in the back, which I listed as F1608 or 2638. Or you can do a correlation study between physical tests and the microbial barrier. The problem with that is a lot of times the correlation testing between the two ends up being a lot more money than just doing one of the little microbial tests. So I think that's probably one of the reasons why a lot of people just opt to do it rather than spend the time to do the correlation and make sure that they're accurate.
Okay. Another question from the audience. Can the same false positive result be obtained in a dye test?
I would say no, because at that point, you're specifically looking for the dye to flow through the seal via a channel. If the dye was migrating and not through a channel, it would be considered wicking. That's another phenomenon that when you're dealing with the dye migration test that you have to be very aware of, because the current recipes that are in allow for wicking within 5 seconds. So there is a time limit on how long you let the dye set against the seals, and it becomes very difficult after that for you to identify true failures from false failures because of the wicking of the fluid through the porous material.
Okay, let me apologize in advance if I mispronounce this word, but shall an accelerated aging test be performed under Arrhenius equations, or are there any other ways to perform accelerated aging?
So the ASTM F1980, which is the recommended standard and the FDA consensus standard, does follow the Arrhenius equation. So again, if there are other options, you can use them. I would definitely suggest that you run the information by a reviewer, maybe like a Q-sub or something like that, just to make sure that they're willing to buy off on the approach. As long as the justification's there and they're comfortable with it. But again, the easiest and approved method is just to follow ASTM F1980.
Okay, another on the longer side. The answer wasn't clear for accelerated aging. So if environmental and shipping tests show no negative effects, do samples from these tests need to be submitted to accelerated aging or real-time aging, or can new samples be submitted to accelerated real-time aging?
Okay, so all the testing that we do is destructive. So it would be new samples would be submitted to accelerated aging. There are only a few tests in the industry right now that are non-destructive. So, and those are really more geared around rigid containers versus pouches. So we would use new samples. Okay,
another question for you. What constitutes worst case SBS/protective packaging? OQ limits, 2x sterilization, etc.
That's a good question and a very, very long answer. Very quickly, everything that the question drives around is to be considered in establishing a worst case. You do need to incorporate the device. You do need to look at the sterilization. You need to look at the pouch, how much sealable area. The document 16775 has a really great annex in there also that outlines some great approaches and thoughts behind how you can establish that worst case packaging system.
Okay, fantastic. Next question. If the product is sold in market as non-sterile and the manufacturer is getting the product as a finished good from a supplier, does the product packaging need a validation?
So it needs the minimum testing that I mentioned, where you're verifying the integrity and the cleanliness. So what you're doing is and I would also probably look at the shipping too. The last thing you want to do is make this product and then send it to your client and then it gets there in a non-usable state. And this is one of those things where market is driving the testing, whereas not the standards. So, and the MDR was specific about ensuring that even if it's non-sterile, those two items do need to be addressed. And I think when it comes to the FDA, that's where they're saying market type issues drive the test.
Okay, great. If your product is humidity sensitive and you must do real-time aging for RH. I'm not sure what that stands for. What is the recommended method? And a second part question, if you do not have access to multiple humidity-controlled storage chambers, would the use of saturated salt containers be appropriate for maintaining RH?
Okay, let's look at the first. If your product is humidity sensitive, so it doesn't matter that your product, whether it's humidity sensitive or it is not, if your product is sterile and sold in a sterile state for a period of time, you do have to do real-time aging. As far as if you're asking for RH, like are you supposed to do testing for humidity as part of real-time aging? The requirements around real-time aging are very vague, and that is definitely becoming a point of question as we move forward in the industry and our testing starts to develop, as we're getting all these new materials that are sensitive to humidity, or in some cases they need humidity to maintain their properties. This is definitely one of those aspects that are going to be taken and evaluated as part of accelerated aging in the future. Where did it go? You do not have access to multiple storage containers. Would the use of a salt-saturated container be appropriate? That's a good question. I'm not quite sure. I think that would fall again into how you justify what chambers you use. The important parts to the accelerated and real-time aging are that you know what the temperature and the humidity are over the duration. I'm sure there are novel ways of monitoring and achieving that, but I don't know that I've seen anybody use salt chambers to do that process.
Okay, and we have another follow-up, actually, I believe. So to clarify on the accelerated aging samples, in the environmental distribution testing, you may include all samples that are also needed for the accelerated aging. Can these testing be totally separated, or the samples subjected to accelerated aging should be preconditioned in the environmental distribution cycle?
Sure. Let me try this from a different approach. So a life cycle product approach is that a manufacturer makes the product, they package and sterilize the product, then they are shipped through the environmental conditions and the hazards, and then they're received at either the hospital or the end user. Once they're received, they can either be stored for a period of time or they can be used immediately. What we do is follow that plan exactly. We take your test samples and we expose them to the conditioning environment and the shipping hazards. An aliquot of those samples is removed, and we verify that there was no impact to the hazards or the conditioning on the devices. If everything is good, the samples that are left from the original testing, the shipping and climactic, then move into accelerated aging, and we pull sample sizes at interim time points throughout the stability period. And we evaluate them for new damage that could have occurred. This is a worst case and is very representative of what your product is seeing. Hopefully that clears it up a little bit more.
Okay, and if not, they have your email address right there in the slide. They can follow up. Another audience question. You state that bond strength is one of the 3 tests that you must have. Besides ongoing process control, what value does completing bond strength evaluation have when bubble leak and dye penetration tell you if you have a sterile barrier?
Let me make a correction. Bond strength and seal strength are completely different things. I'm specifically talking about the seal strength of a peelable pouch. Um, I'm sorry, the question disappeared off the screen. Can you repeat the last part of it?
Sure, let me just pull it up.
Sorry.
That's all right. That's all right. Besides ongoing process control, what value does completing bond strength evaluation have when bubble leak and dye penetration tell you if you have a sterile barrier?
So the bubble and dye leak are specific for one characteristic. They are only verifying the integrity of the package. They have no reflection of the strength that those seals have. So you also do need to identify that characteristic because if your product is too heavy and your seals are too weak, you're not going to make it to the facility where you sell your product. But if the seals are too strong, you're going to pass an integrity test, but the end user, when they open that package, are going to launch their product across the room because they can't open it. So that's why you're looking at both the strength and integrity as part of an entire focus around that sterile barrier system.
Okay, fantastic. All right, one more, Wendy, I think, and then we can let you off the hook. So the last question we're going to have. You mentioned a test method validation guidance. Can you clarify which guidance it is?
I don't know that name off the top of my head, but I do have the email for the person asking the question. I can directly send that information when we're finished. Sorry.
That's all right. Perfect. That works out well. So as I said, unfortunately, that's all the time we have for today's presentation. I'd like to thank our speaker, Wendy Mach, for being a part of this webcast. I'd also like to thank our sponsor, Nelson Labs, for its support. Finally, I'd like to thank you, the viewer, for attending this session and for asking so many questions. I hope you found it to be a valuable experience.